All right, all, welcome back. The second part to this kind of beginning lecture is I want to kind of tie the characteristics of life and what we're going to, things that we're going to build off of. This is like the base layer. So knowing how the different properties of living organisms versus non-living organisms, and kind of those properties. We'll get into cells in the next series of lectures, but really this is kind of just an overview. If you're struggling with knowing the characteristics of life, you will need to cover this material by reading through it quite a bit. This should all be simple review coming out of a introductory bio class. Okay? So what are some of the characteristics of life? First of all, they have organization. And so that organization, it can be, we're talking about cells, and cells making tissue, tissue making organs, but we could be talking about single cell life, where that organization is not past the cells, but rather it's the organization of the organelles that are in that cell. Maybe we're talking about the nucleus, the smooth and rough endoplasmic reticulum, the gold. Oh, wait, we could be talking about single-celled life that doesn't have organelles. And then we're talking about, well, now we might be talking about organization at the protein level. Okay, all living organisms have organization. Now, the hierarchy or the level of organization differs by a lot of different organisms, but in the field of anatomy and physiology and the related fields that you might be going into, whether that be allied health, nursing, forensics, etc., you're going to deal with all types of organisms. You're going to deal with those bacteria which don't have organelles, yet they have organization when it comes to protein function, when it comes to the structure of their proteins, when it comes to how they go about consuming material and generating energy. And so we'll come back and talk about that when we talk about different diseases. And for those of you who are continuing after anatomy and physiology and get into micro, you'll talk a lot about those organization, etc. Cellular composition. Okay, so we know that characteristics of life, you must have at least one cell. Okay, so according to the laws of what's alive on this planet, you must have at least one cell. Okay? Metabolism, have to have the ability to convert something into energy. Okay, and so we'll talk about that conversion. We'll talk about cellular respiration. We'll talk a little bit about fermentation. Okay, and that will be, for most of you, hopefully a review. You have to be able to respond to stimuli. So you have to have some kind of sense. That sense could be like visual, like us, actual eyes, touch, hearing, smell. Okay, those are all senses. But from a cellular perspective, it could be the sense of not enough cytoplasm, make more. Membrane has, a puncture in it, make more phospholipids. I don't have enough protein channels to allow glucose in, make more GLUT4 protein channels. So it's a stimuli, it's a response to something, okay? Some reaction to a change in the environment. And then movement, we often talk about movement from a perspective of the entire organism might be able to move, okay? But movement in general, it could mean that we're talking about substances within the organism. Okay? If we're talking about a stationary organism like a bacteria that doesn't have a flagellum or cilia or ability to move, maybe it still has movement though, because it's generating proteins, and those proteins, they are hooking on to microtubules, and they might be migrating from inside portion of the cell out towards the outer membrane. Okay, that's still movement, that we'll often talk about. Probably won't get into too much of that in this course, but maybe in micro. Okay, some other things that are really important. Homeostasis, or maintaining some stable internal conditions. Now, homeostasis means something different for a lot of organisms. We happen to, from a homeostasis perspective, we happen to often think about, well, there's this single number and this right kind of number, like our for example, the temperature of our body, 98.6 degrees Fahrenheit, and deviations from that means we're outside of homeostasis. Okay? But not all organisms have that kind of homeostatic uh, truncation. So they're not all organisms are so narrow when it comes to homeostasis. We're not narrow when it comes to homeostasis for other things. Different hormone levels, different glucose amounts in our body, different sodium levels, the amount of water we have. Okay, so we have a lot of flexibility in some things, and other things we keep it fairly rigid. Okay? Development, so the change of a form or function over time. Okay, so differentiation from one cell type to another cell type. Okay, aging, growth, all of these things have to do with development, and we'll cover some of that. Okay, of course, the ability to reproduce is very important. You have to be able to pass on genetic material on to the next generation. Now, it doesn't have to be sexual reproduction. It could be asexual reproduction. Again, when you get into micro, you'll talk about lots of asexual reproducing organisms and their ability to pass their genetic material on to the next generation. And then, of course, if you have reproduction or genes that are being passed on, those genes have to be able to change over time, mutations, and some of those mutations have to cause alterations, and population has to change over time. Right? And that's what evolution is the genetic change of a population from generation to generation. Okay? And of course, all the features, all the structures that we'll talk about from an anatomy perspective, from a physiology perspective, all of them are the result of evolution. And we can point to the similarities we have in our ancestral species and how their anatomy works and their physiology works. Okay, physiological variation. Okay, this is important. We'll often hit this when we talk about case studies. Okay, we'll talk about sex variation or age variation, the diet, the weight, the physical activity, genetic information that might tell us the environment that individuals might find themselves in. All of these can have a direct effect on that homeostasis that we're often searching for. All right, so two like typical physiological values. Okay, so we can look at, again, often I will present this to you when we first look at a vital. You might say, okay, in the average male respiratory rate is this, and the average male heart rate is this. And so we'll look at these. And this is just an example of that. A 22-year-old male at 154 pounds, his average calorie intake should be about 2,800. A female, okay, at 22 years old, 128 pounds, average calorie intake maybe 2,000 kilocalories per day. Okay, difference in physiological values, difference in the ability to burn through the calories. Okay, there's just a difference in the sex of the individual can cause differences in the number of calories needed for the individual. That's important to know, given that, if you're constantly, giving an individual 2,000 kilocalories per day, and that individual's a male, you might be under supplying them the energy they need to recover. Same thing though, if you provide 3,000 kilocalories for a female, maybe you're oversupplying them calories, and those extra calories are just going to go into fat storage. And so there are ways at which these play a direct role. Okay, and variation can change over time. Okay, and variation can definitely allow us to make mistakes. Okay, if you're not considering variation, then you might over medicate women and under medicate men. Okay, or you might make mistakes that are based purely on the sex of the individual. You could be making mistakes that are based purely on the age of the individual, okay, the weight of the individual. So all these things have to play a role. And that's where we start talking about the difference between homeostasis and what we call allostasis. Okay, so homeostasis is definitely kind of this range that we typically have. Like, this is the normal range, and majority of humans fit in this normal range. So normal body temperature range of individuals is like 98 to 99, somewhere in there. Okay, and so those are the normal ranges. Outside of that, you either have a fever or your body might be too cold. Okay? And you have ways at which you can regulate that. Okay? Well, there are some situations though where if you have chronic stimulation, or you have chronic increases in things like hormones, or maybe glucose levels, or there's lots of other examples, and we'll talk about some of them. Okay, you may deviate from that homeostasis by getting a new set point. Okay, so your set point might increase over time if you're constantly stimulating. Okay, so stress hormones, for example, if you're constantly stimulating yourself because you're constantly being stressed, your set point will start increasing potentially, or decreasing, depends on what we're talking about here, but it could increase and just keep increasing till eventually you're not getting a reaction in your body to those stimuli, to those hormones, until you have a much higher dosage of that hormone. And so, because you have a new set point, and that's called allostasis, okay, or the difference between the original set point and the new set point is called the allostatic load. Okay, and this happens often. It happens with medication. Okay, so things like pain tolerance, or pain reaction to pain, or the ability for the medicine to work, often will set a new set point. So an individual that might take Tylenol to deal with headaches, if they constantly are taking that year after year after year, for some individuals it may take many years. For some individuals it might take a single year, a couple months. They might set a new set point to where now the single dose that's on the bottle, it's not doing anything. Okay, because that stimuli, they need more. Okay, and we'll come back and we'll talk about these stimuli and how that works. Okay. All right, negative feedback, homeostasis. Again, we talked about homeostasis before. Okay, homeostasis is trying to maintain, okay, that internal condition, and triggering the ability to maintain it. So, for example, if you have too much sodium in your body, Maybe you become thirsty, so you're trying to dilute how much sodium is around. Okay, so these are homeostasis or activities that are trying to stabilize conditions within the body. Okay? If your body's too hot, okay, you tend to sweat, and so evaporative cooling might try to lower the temperature back down. Okay? And so Claude Bernard came up with this based on temperature, okay, and really that we have fairly constant internal conditions, okay, and he was really looking at temperature. And then Cannon later described this process of our ability to thermal regulate as being a homeostasis, maintaining that internal temperature. Right? Temperature just happens to be a great characteristic in humans and really all endotherms, because their temperature doesn't fluctuate very much. And so the allostasis set points don't really raise very much, from a homeostasis perspective. Had they looked at something like glucose level or salt level or something like that, which has a much wider range, it would have been a lot harder to figure this out. But nonetheless, based on homeostasis, you can also have what we call negative feedbacks. Okay, so that means that if you're generating too much of something, that can fit back in and stop the system. Okay? A lot of our hormones are based on negative feedback loops. Okay? A lot of systems that have a very narrow range. When you talk about their normal range, they'll have negative feedback, so they don't get out of whack too easily. So negative feedback is really important, for the perspective of it stops the process from continuing. Okay, it's from going too far out of whack. Okay, and so this, though, like I talked about before, this can be compromised if that stimuli happens constantly over time. Then you can get a new set point. And it depends obviously on what we're talking about when we're talking about negative feedback. Okay. But you can get this new set point, and then the system is going to be ramped up or much lower than what you might expect or what you might normally find. And I'll show you a bunch of examples of this as we go. Okay. So one thing when it comes to negative feedback, again, most of the time it's fluctuating within a limited range. Okay. And so the negative feedback is the mechanism to keep the whatever we're talking about, the temperature, the hormone, the channel, the potassium level, to keep it as close to the set point as possible. Okay. There are other feedbacks which we call positive, and so you can have both a negative feedback loop. Okay, and it's just a mechanism. It normally means that you have a biochemical pathway. Biochemical pathway, just a reminder, is one thing, okay, is used. So one product is used as the reactant in another reaction. So you have a product, reactant, new product, reactant, new product, and it's a pathway, a biochemical pathway. Because it occurs in a biochemical pathway, we often have loops where maybe the last product will fit back in to the original protein in most cases, and it will stop the process from happening, from occurring. Okay, so when you have too much of a hormone, that will slide back in and stop the process from occurring further and producing more hormones. Okay, so Homeostasis of body temperature is a good example of kind of a negative feedback loop. Okay, if you're too warm, okay, the blood vessels will start to dilate. Okay, so they start to increase in size. Okay, so vasodilation occurs. Okay, if you're too cold, okay, vasoconstriction occurs. Okay, so you could go too warm, vasodilation, and sweat. Okay, so this dilates, it moves the volume, it pushes, and so it allows the blood cells to get closer to the surface, and now the surface is wet, and you get evaporative cooling, and so the blood cools. If you're too cold, you don't want those vessels close to the surface. You want them as tight as possible, constricted, okay, away from the cooling mechanism, and you have vasoconstriction. Okay? And so you have both a heat loss mechanism and a heat gain mechanism. And so here, this is just showing temperature over time. Again, about 98.6 or 37 degrees Celsius is the set point for a majority of humans. Okay, you get it to about 99.5 Fahrenheit, and you'll start to get vasodilation. The individual will start sweating. Okay, if you get below about 36.5 Celsius to about 97.7 Fahrenheit, the individuals start shivering and you get vasoconstriction. Right? And so these mechanisms are negative feedbacks. They are, as the temperature is rising, okay, the feedback is to stop it from rising and push it back to the set point. As the temperature is decreasing, the feedback is to push it in the opposite direction. That's where the negative comes. It goes in the opposite direction of the trend. If it's trending to a hotter body temperature, opposite, go back to the set point. Colder, opposite. Okay. Positive, on the other hand, is a little bit different. Okay. Let's look at another example of homeostasis when it comes to blood pressure. Okay, so when you rise up out of bed, okay, the blood drains from your head and the pressure changes. So your blood pressure falls in that region. You have baroreceptors, which we'll talk about a bunch, that occur in different regions of your cardiac system. Okay, your aorta, for example, has baroreceptors in it, and that tells the body, okay, the brainstem, the medulla oblongata, it tells it that the pressure is either decreased or increased in that region. Okay? And then based on that, we can get a new signal to either, in this case, increase your heart rate, or in other cases, maybe decrease your heart rate. Okay? So if that cardiac center transmits the signal to the heart to increase the heart rate, okay, pump more blood, that was going to raise your blood pressure and it'll restore homeostasis. Okay? So you can see here, you have baroreceptors that occur like on the aortic arch, okay, that can signal the pressure's too low. Okay? So it signals the medulla, okay, and then the medulla will send the signal back to the SA node, okay, and that will speed up the heart. And so the heart rate will increase, okay, and that will get you back to homeostasis. Okay. So again, this is kind of normal feedback loops. This is kind of what's involved. You have some type of receptor, a sensory receptor, and this could be on the outside of your skin, sensing temperature. Okay, it could be sense of pressure, it could be sense of taste, smell, vision. You have some type of receptor that's going to sense a change in either your vision, your hearing, your taste, your touch, whatever it might be, or in this case, pressure, then that signal is going to be sent to some type of control center, right? It could be a control center that is nearby. It could be a control center that's in the brain, in the medulla. Um, it lots of different regions, and then you're going to get a response, and that directs a response to increase, decrease, whatever it might be, depends on what the receptor is receiving, what kind of signal is it sending. Okay, and then that signal is going to be sent back to the effector, and the effector will carry out the necessary, in many cases, the necessary response to restore homeostasis. All right, and finally, look at positive feedback. Not a huge amount of examples of positive feedback, but there are a few that when you start down a pathway, instead of stopping the pathway, it continues the pathway. And it starts moving in the same direction to a greater deal. So it's not a negative feedback, it's not a correction action. It's a inline action. Right? And so some examples, blood clotting is a good example. So once thrombocytes start to form a clot, they send out signals, okay, and those signals change proteins in the blood to form kind of a fibrin and net. And so this is a positive feedback. Okay, it's continuing the process. Okay, childbirth, a great example of this comes from childbirth, where you might have the head of the fetus is pushing up against the cervix. Okay, there's nerves that are in the cervix that then transmit a signal to the brain. Okay, that then the brain is stimulated to produce oxytocin, and then oxytocin, okay, is used to cause the uterine contractions. It's used to cause the muscles of the uterus to contract and push the fetus towards the cervix. Okay? It's also related to lactation or milk production, and we'll talk about that as we progress. But these are positive feedbacks. Now, sometimes they can be dangerous. Okay, so you don't have a negative feedback, you don't have a corrective mechanism, and it can keep going in one direction to a great deal, and that can be problematic. So you can have things like a runaway fever, where you might have a virus that has induced, or your body's inducing a fever because of a viral infection, but there's no controlling mechanism. There's no negative feedback. Okay, the cytokines just keep on producing the stimulus of produce higher temperature, and we can get what we call a cytokine storm that occurs, and then you can get a runaway fever, which is very dangerous. Okay. Other things, okay, gradients and flow. Okay, we'll talk a lot about gradients, whether that be a temperature gradient, pressure gradient, chemical concentration gradient, charges when we're talking about action potentials and things like that. But there's a lot of different gradients, and often they're working on the kind of negative feedback loops. So you have a change in the gradient. Something's going to, some kind of receptor is going to pick that up. Signal is going to be sent. Control center will send a signal back to the effector, and you'll get a change in that gradient. Okay, so pressure gradients, okay, chemicals, kind of probably talked about this many times in previous classes, but most of the time when we're talking about gradients, it's flowing from a high concentration to a low concentration. So it flows from high pressure to low pressure, flows from high concentration to low concentration, flows from one electrical charge to another electrical charge. Okay, so for the most part, it's flowing from high to low. Okay, and so you get mostly what we call down gradient flow. Okay, so down the pressure gradient, down the chemical gradient, down the charge gradient, down the heat gradient. Okay, you can get up gradient movement, but normally that results in spending energy. And we'll talk about some situations where that happens. Okay, so here you can just see some of those gradients. So high pressure to low pressure. So the highest pressure is going to be right at the aortic arch as the blood is leaving the heart. Okay, and low pressure, the further you get away from the heart, the lower the pressure. Okay. Chemical gradient, high glucose level, maybe in the bloodstream, is flowing into cells that have lower glucose levels. Okay. When we're talking about sodium, potassium channels and things like that, so ion channels, you got high ions on the outside flowing in to low ion concentration. Heat from inside flowing out to cold air on the outside. Okay? And so these are typical gradient channels. All right, and I want to finish with a little bit about terminology. So the terminology in anatomy and physiology, it's what I often will call mixed terminology. Sometimes you have old terminology that still is around, and then sometimes you have new terminology. And so more modern terminology is to use terms that relate to the structure or the function of the object that you're talking about. Old terminology might have the names of individuals who discovered that anatomy or that physiology. And so you can kind of get a mix of the two. Right? And so eponyms still reside on structures. I try to provide, when we talk about these structures, I try to provide kind of both and a new name, and then the eponym, the name that is often used because it's related to the individual who discovered this structure. Okay, we have a worldwide kind of naming convention, and for the most part, it rejects the eponyms, but I'll tell you that they're still used, they're often used, and if it's going to be used by, a medical facility, I'll try to make sure that that what that eponym is. Okay, but for the most part, we prefer to use Latin names, and those Latin names have actual value to them. They actually mean something, and they have context often. Okay, and so for the most part, in most countries, okay, the TA or terminology, anatomonica, is the standard, and most countries actually use this. Most countries have kind of provided that. At least most countries that the majority of you might work in have kind of accepted that. Okay, and that really brings me to the last piece that I'll often get questions about. Well, why are we graded on spelling? Why do I need to know how to spell these words? Well, for many cases, it's very important, because being just a single letter off can mean that you're talking about a different structure. You're talking about a different medicine. You're talking about a potential different procedure. Okay, and so it's really important to maintain safety and precision by knowing how to spell the terms correctly. And so spelling does count, especially on lab practicals. Spelling counts. In lecture, it's got to be close. Okay? You'll be writing essays and short answers and things like that. And so if you misspell a word by a single letter in lecture and there's context to it, okay, that's different. It's different because now I know the context, I'll know what the term is. But if it's just a blank on an exam, like the name of this tubercle on this bone, and you misspell it, well, there's no context to that, and it's hard to decipher what you meant by that. Okay, and that's really why the importance of spelling. Okay, all right. Next time out, we'll talk about cells.